Hi, I'm Maddie O'Hare.
And I'm Franklin Leonard, and today we're going to be talking about Mycobacterium leprae and its effect on Schwann cells.
So we chose this topic because we read a book last year in Anatomy and Physiology that talked a little bit about leprosy, and we were really interested in that and wanted to dive deeper into that topic. Mycobacterium leprae targets unmyelinated Schwann cells more frequently than myelinated Schwann cells.
This is because the myelin provides both a physical and physiological barrier. Nerve biopsies have shown that Mycobacterium leprae more frequently target unmyelinated Schwann cells, and it is actually very rare to target myelinated Schwann cells.
Next, we're going to be looking at some data from a study comparing demyelination percentages between Mycobacterium leprae as a whole, just the cell wall, phenolic glycolipid 1, and phosphate buffered saline, which is the control. So phenolic glycolipid 1 is a biologically active component of the Mycobacterium cell wall. And when we look at the graph over here, you can see that the Mycobacterium leprae as a whole caused the greatest percentage of demyelination, and then the cell wall caused the second greatest amount of demyelination. The third was phenolic glycolipid 1, and lastly was the control, which was phosphate buffered saline. And all of this was done in the absence of immune cells, and basically shows that Mycobacterium leprae may cause demyelination.
So now we look to a study called Myelin Breakdown Favors Mycobacterium leprae's Viability and Survival in Schwann Cells. This study was written by Bruno Sequeira and numerous other authors. And in this study, they pharmacologically inhibited myelin degradation in Schwann cells using a J&K inhibitor. Now this J&K inhibitor prevented the degradation of myelin in both cells infected and non-infected with Mycobacterium leprae. They then measured the Mycobacterium leprae pathogen content in cells both treated and not treated with the J&K inhibitor. As we can see from these graphs over here, the cells with the J&K inhibitor had much less myelin breakdown than the ones without the inhibitor. Now, in the cells that were treated with the J&K inhibitor, there was a decrease in myelin degradation. And because of this, there was 3.3 times less Mycobacterium leprae content found in these cells. This showed evidence that myelin breakdown is a main contributing factor to Mycobacterium leprae's persistence in myelin Schwann cells. As we can see from this graph over here, the black bar is the Mycobacterium leprae infected Schwann cells, and its height is the Mycobacterium leprae content. On the right, it is the same Mycobacterium leprae infected cells, however with the J&K inhibitor. With the decrease in myelin degradation, there is an obvious decrease in Mycobacterium leprae pathogen content.
So, for the leprosy disease to progress, we need unmyelinated Schwann cells. With only myelinated Schwann cells, the disease would not progress very effectively. So, to do this, the Mycobacterium leprae basically goes within the Schwann cells and causes the unmyelinated Schwann cells specifically to go through the cell cycle more often and quicker, so that more unmyelinated Schwann cells are produced, and that allows the Mycobacterium leprae to infect more cells and spread throughout the body more and more efficiently than it could if only myelinated Schwann cells were within our bodies. So recent studies suggest that the myelination differentiation process in Schwann cells is due to a balance of positive and negative regulators within the cell. If it is a more positive balance, then it's going to be myelinated, and if it's more negative, it will be non-myelinated Schwann cell. So this is important to recognize because in vivo, when a Schwann cell is injured, it can cause demyelination, so it's shifting that balance from more positive to more negative. This also happens when Mycobacterium leprae infects the cell, because it is injuring that Schwann cell and causing the balance of the regulators to shift more towards the negative side than positive, and therefore causes demyelination. Pathogenesis of infectious disease is greatly aided by its ability to target one specific cell type. So for Mycobacterium leprae, this is the non-myelinated Schwann cells. So when they can't find those non-myelinated Schwann cells or they have a lack of them, they go to causing demyelination by switching up those regulators and changing them to more negative, which causes demyelination, or they have the non-myelinated Schwann cells go through the cell cycle more often so that they have more non-myelinated Schwann cells to infect.
To put in simple terms, Mycobacterium leprae targets unmyelinated Schwann cells because they are much easier to infect. When you decrease the amount of myelin degradation, the amount of Mycobacterium leprae pathogen content decreases as well.
We hope that you enjoyed listening to our presentation about Mycobacterium leprae and the leprosy disease. I hope you learned a little bit more about it and are interested in learning more about infectious disease. This was super interesting for me to learn more about. It was a super interesting topic, so I hope you enjoyed it as much as we did. Thanks, guys.
Thank you.
I guess